Good morning, all. Hello. Right, today topic is kinetic of DNA reassociation. Kinetics of DNA reassociation, or another name is called as Cart curve analysis or Cart value. This is also important for short answer. So last class we discussed about C value paradox, genome organization, chromosome structure, all this. And these all are important. And another important topic is today Cart value. Cart value or DNA reassociation or Cart curve analysis. Right. So first let's discuss what is the definition Cart curve. The simple definition: the product of the original concentration of denatured DNA and time, and giving a useful index of the DNA renature. That is called Cart value. The renature has occurred which can be used to estimate the length of the unique DNA sample. Simple, for example, in this, we should clearly discuss about what is denaturation, what is renaturation. The simple what we analyze is in this topic, the time of renaturation, reassociation or renaturation of the DNA. That is estimate about the cat value. The first, how denaturation occur in DNA. Let's see what is denaturation first. Denaturation, denaturation, so one, correct. For example, this is DNA. DNA double strand. DNA double strand. When you eat this DNA, eat, when you apply the eat DNA, what happen? DNA strands get separated. DNA strand gets separated into two strands. This is one strand, nucleotide sequences, here nitrogen bases, and here different nitrogen bases. Separated, both strands are separated. So this is called denaturation. After denaturation, after getting cooled down the heat, when you reduce the heat, when you reduce the heat, when you reduce the heat, now automatically these two strands again renaturation, unite, reassociate, again reassociate. And we should take the time to reassociation. In what time they reassociate? After denaturation, when they renature, when they renature. For this renaturation, mainly the DNA content mainly depend on, this is important, mainly depend on concentration of DNA, concentration of DNA, concentration of DNA, DNA concentration, first of all, DNA concentration, and based on the time and buffer factor. These three, may the vital role in the renature of the DNA. So these three are directly proportional to the cut value performance. For example, here if we take the number of concentration DNA, here what happened? For example, if you take the graph, this is genome reassociation percentage. Genome reassociation percentage this side, and right. Correct. If you take the cut value mainly depends on concentration of DNA, concentration of DNA, and time and buffer solution, and time and buffer solution. These are the major important factors in the cut value analysis. Let's see if we take the graph. This is cut value X. It's taken cut value COT cut value, and here single standard DNA percentage. Single standard DNA percentage. Single standard DNA percentage on the side. Genome reassociation percentage or single standard DNA. Here the which DNA reassociated, double standard DNA or single standard DNA. Single standard DNA get reassociated, right? When we take the single standard DNA percentage, this is mainly if you observed in Escherichia coli, E. coli. What happened in E. coli? Then the first, when the single stranded DNA content in high, automatically reassociation process dramatically increase like this. And while the single stranded DNA, for example, here the 100 single stranded DNA present. But what happened during reassociation occurs? What happened during reassociation occur? Automatically the single stranded DNA becomes double stranded. That means the single stranded DNA, single stranded DNA percentage decrease or increase. Automatically, the single stranded DNA percentage gradually decrease. Gradual decrease. That's why this graph shows like this. The completely decrease, and finally, right. So this curve called as sigmoid curve. This curve called as sigmoid curve, and in this mainly depend on actually here the first in single stranded DNA also high repeated DNA present in first which DNA renatured high repetitive DNA. I will explain later what is repetitive DNA, moderate to repetitive DNA clearly. But here remember, first in a medium, if you add the single stranded DNA percentage and it would give appropriate time and also buffer medium, buffer solution, what happen? Our concept is first of all denatured DNA renatured. How denatured DNA renatured and how much time taken? These are we need to be observed, right? First of all, how the double stranded DNA renature already we discussed. Double standard when you apply the heat, what happened automatically? The double stranded DNA gets separated into single strand. After get cool it down, these two again renatured. This time we calculated the renature time, and for this renature, the mainly it is depend this renature process. This renature process mainly depend on the content of single stranded DNA. The more single stranded DNA get more reassociated, right? More single stranded DNA get more reassociated, and also you take more time, and also the reassociation percentage also increase. If you increase the buffer medium, automatically reassociation increase. Just a minute, just a minute. Sorry for the disturbance. Right. So right anyway, so this renature mainly depend on the single standard content, time, buffer solution. These three the major, and viscosity also, but this first three is the major one. So just the- when the single standard concentration present, for example, here the 100 single standard DNA present, but in this 100 single standard DNA, first high repetitive genes. I will explain later what is repetitive genes. For example, if you take this DNA strand, one of the nucleotide sequences repeatedly formed. This is high content. The first the high repetitive genes for this. Let me change color. The first high repetitive genes reassociated, right? Later, let's change color. Later, what type of gene reassociated after high repetitive, moderately repetitive genes for this region until here, this curve only moderative, 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 and what happened? Let's do it. 1 minute. Right. Mainly this experiment done in bacteria, prokaryotic organisms. And if the same experiment is carried out using DNA purified from the complex eukaryote, such as human, then a sample sigmoid curve is not produced because the genes are very complex and also very and also the repetitive DNA and moderate repetitive DNA and also I forgot one thing. After moderate repetitive DNA, next renatured DNA stands are renatured DNA stands are, let's change color, and less repetitive DNA, green color, less repetitive DNA, less repetitive DNA, right? For example, within the 100 single copy, within the 100 single copy, 30 consisting repetitive DNA strands, 30 strands repetitive DNA strands, 10 moderate, and remaining 10 low. The first of all in this 30 single copies reassociated. Next, 10 single copy reassociated, and later the last this is taken the high time. Is taken the more time to reassociate it. In this context, right? In this context, first of all, the high repetitive DNA immediately reassociated. Now some of the content, now the single stranded DNA completely reduced. And again, the remaining moderate DNA also reassociated. And again, the more DNA strands decrease. The final low repetitive DNA, now the all R gets reassociated. Hello. Good morning all. Why are you joining late? Hello. Vijay Sahi, Saleha, Swati, audible to you?
Yes, sir. Audible, sir.
Right. So once again, let me give the brief introduction about the cat value because without concept you can't understand anything, right? First of all, in cat value mainly depend on denature and renature. First of all, the denature. If the double stranded DNA, for example, this is double stranded DNA, the double stranded DNA, when apply the heat, automatically it is get it is denatured. Double stranded DNA, when you apply heat, it is denatured. Later, it becomes single stranded copy. After that, when the heat cools down, automatically again these two single stranded DNA renatured. This renature mainly depend on single stranded DNA percentage and also time and also buffer solution. Buffer solution. If you take the graph, if you take the graph, y-axis single stranded DNA percentage, y-axis single stranded DNA percentage. Before that, you should remember
Rama.
am I audible? Hello? 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 Swati, Nagarani, Saleha. Am I audible?
Yes, sir. Good
Right.
morning.
Right. Sorry for the inconvenience, some technical issues.
Good
Right.
morning, sir.
Right. Good morning. So right. So as I said earlier, here the cat value mainly depend on single stranded DNA percentage. Now have taken the y-axis single stranded DNA, and time which is in the buffer medium, and now the cat value. Now they are taking cat value percentage here. Cat, what is cat value? Already told you, cat value means denatured DNA, renatured. That time is called as cat value, right? Now on y-axis, have taken the single stranded DNA percentage in buffer medium and give the appropriate time. Now in this time, it is experiment in Escherichia coli genome. Now what happened first? In the first, highly repetitive DNA reassociated, later moderate DNA reassociated, the finally low repetitive DNA reassociated. If you take high repetitive DNA, it is represented with red color. So red in color, this is high repetitive DNA. Next green color, moderative, and final low, low repetitive DNA. Now reassociation completely increasing here. The first in starting 10 minus 3, here only single stranded DNA. Now tell me the single stranded DNA percentage decreasing or increasing? Decreasing and increasing. When give the proper time and buffer solution, automatically what happening? The single stranded DNA percentage decrease or increasing. This graph shows decreasing single stranded DNA or increasing single stranded DNA. This graph shows here the cat value, increasing cat value. Increasing cat value means what? Increasing deassociation or reassociation of DNA. Hello? Thirty repetitive DNA, 10 moderate, 10 low. In this first 10, immediately reassociated. Then how many single copies remain? How many single copies remain? Hello? The total 100 DNA single copy, 100 DNA single copy. In 100 DNA single copy, 30 high repetitive DNA is 30 copies high repetitive DNA. This consisting high repetitive DNA. Now this 30 reassociated. Now how many remain? Hello? Hello?
Yes, sir.
How many remain? For example, I have taken the single standard DNA percentage 100 copies. In 100 copies, first, as I said, first repetitive DNAs reassociated. It follows moderate repetitive. It follows low repetitive DNA. Now tell me the first, when high repetitive DNA, already given the number high repetitive DNA stands 30. If the 30 high repetitive DNA stands each other reassociated immediately. Now, okay, tell me one thing. Now the 100 single stranded copy number decreasing or increasing?
Increasing.
That's why I request you, please join immediately after when the class started, join immediately. If you join lately, there is a chance to missing some of the important things. The first of all, what is cat value? What is cat value? In our book also, the page number 15, 1.7 kinetic of DNA reassociation. Of course, given in our material, but it is some of complex. Now it is make it very simple. So first of all, here the remember one thing, the kinetics of DNA reassociation or cat value association, the any mode of question, any the same, they are the similar words. In examination point of view, if they ask kinetic of DNA reassociation, or write about cat value, these are all same. So first of all, what is cat value? First, any question you should start with definition. What is definition cat value? Just now I explained. Cat value means this is double stranded DNA. Are you able to see? Are you able to observe this slide? Marker. Hello? Don't be passive. Please active. Hello? Please respond.
Yes, sir.
Swati, Nagarani, Saleha, Santosh Kumar.
Yes, sir.
Are you there?
Yes, sir.
Yes, sir.
What happened? Why are you disturbing like this? Hello, BRAOU host. Are you there?
Yes, sir.
What happening there? Why are you interconnected? Why are you disturbance? Is it problem in your side or my side? Hello?
Yes,
Hello.
sir.
Yes, sir.
Is there BRAOU host who operate all this? Right. What happening, man? Are able to see PPT slide graph?
Yes, sir.
Right. Final, just remember one word. Cat value means reassociation of DNA. The reassociation DNA from deassociation, denatured. Denature means separated. Double strand DNA separated, denature, again renature. This is called cat value. and I have taken here the single stranded on y-axis. Automatically hello.
Yes,
Hello,
sir.
sir.
Well, some technical issues. Please disconnect and again reconnect. Okay.
Yes. Okay, sir.